Good morning, ladies and gentlemen. Firstly, I'd like to thank everyone for being here today. And I'm also very honored to represent my team, Precisia, in collaboration with GenoByte. And we are a precision medicine pipeline. So by precision medicine pipeline, what I essentially mean is we are involved with the genetic testing, conducting whole genome sequencing, and based on the polymorphisms, we are able to map the polymorphisms to a disease condition if they are of clinical significance, or if they are something that's natural which results in variability, we try to understand what are the implications of the variability. So before getting into the depth of the presentation today, I'd like to give you a very classic example which has led to the evolution of epigenetics in the first place. So it's the example of the two bees, the worker bee and the queen bee. If you look at the literature, both worker bee and the queen bee, they are both genetically identical, which means they have inherited the same genetic makeup. But if you look at the queen bees, they are fertile in nature, they are able to lay eggs, and they are also the aging process in queen bees that's also very slower compared to the worker bees. Now, what could result in such differences if they essentially have the same genetic makeup? So that's where epigenetics come into play. The worker bees, they produce a component called the royal jelly, which the queen bees feed on. And this royal jelly has been said to induce some methylation patterns at the genetic level, which makes the queen bees more fertile in nature with longer lifespan. So that is what we are specializing at our precision medicine pipeline, where we exploit the features of DNA methylation in order to improve the quality of lives of patients. This is an interesting exhibit which the Time magazine published in the year 2010, where during the 18th century in the northern Sweden regions, there was famine which was quite prominent at that point of time. Now, why this is very important is because during famine, there was a lot of population studies done where a group of scientists, they tried to sequence individuals and they have looked at the differences between different generations. So the generation that was drastically affected by famine, the subsequent generations were born with tendencies that predisposed them to cardiovascular conditions and obesity. Now, if the same genetic material is transmitted from one population to another, how does famine exactly, which is an environmental factor, how does it affect the subsequent generations in inducing that predisposition or the risk to obesity? So this is where epigenetics revolution began, where we are trying to explain how exactly your DNA methylation and hyper and hypomethylation can impact or can be remembered for the next few generations, and the offsprings have the risk of expressing them at some point in time. So what makes us unique? It's not only the genetic makeup, because if you look at the genetic makeup, you and I are 99.9% similar. But what makes us unique? They are the polymorphisms. So the book you're born with is your genome, but the book you write during the due course of your life is your epigenome, which is why even monozygotic twins per se, they have certain differences in terms of predisposing themselves to obesity, and that mainly comes down to epigenetics. So we are trying to exploit this factor of epigenetics in order to make sure that during your lifestyle, during the due course of your life, you follow certain patterns in place which helps you live longer and also enhance the quality of life. Now, this is a classic example of how your cosmetic products, your supplements that you consume on a regular basis, and also your MTHFR, the classic mutation that explains epigenetic mechanisms quite well. So your MTHFR mutation, there is a variation, and now this variation stops or prevents the complete conversion ability of your folic acid, which you get from your diet, into its activated version. Now, what could be the implications of this? Under the conventional thinking, where your green leafy vegetables are said to be beneficial, candidates with this mutation, a combination of two different variants, cannot do that 100% conversion. Now, if they cannot do the 100% conversion, the folic acid that they consume remains as folic acid in the system. And as a result of the accumulation, if it's going to be a pregnant mother, it can result in neural tube defects, especially in the progeny. Now, when we are doing a genetic testing, we are able to identify this MTHFR mutation, and accurately we are able to curate a diet plan to make sure that we hypermethylate or hypomethylate certain genes, which will now allow the smooth conversion of folic acid into the 5-MTHF, which is the activated version. And now this we are able to epigenetically modify just with the help of right diet and bioactive components. The same concept goes with the supplements as well, and also some of these cosmetic products that's able to induce that epigenetic changes at the root level. Now, this brings me to the next concept of exposome. So, what is an exposome and how is it intricately involved in upregulating or downregulating certain genes, complying with the principles of epigenetics? Now, exposome is nothing but the day-to-day activities, the day-to-day factors that you are exposed to, be it your diet, be it your exercise, your overall lifestyle comprises that entire circular concept of exposome. Now, based on what you do on a day-to-day basis, be it a simple habit of just getting enough sunlight in the morning, the vitamin D, it can have the ability to interact with your epigenome, and ultimately it can hypomethylate certain areas of the genes, which will then control the way you express certain genes. So your DNA sequences are going to be the same. They are not going to change throughout the due course of your life. But the way in which you express certain sections, that comes down to your epigenetics. So which is what we are trying to focus on, and we are also trying to address some very, very important questions when it comes to the exposome. What are the constituents of exposome, which still needs a lot of research going back and forth to understand whether clinically there are important significance of having that good understanding of your exposome and genome interaction. What are the constituents of your exposome and what are the health implications as a result of having certain factors you're exposed to during the due course of your life? Now, your exposome is your writer and your eraser. In the sense, in very, very simple terms, the factors that you're exposed to during the due course of your life, it has the ability to shut off the expression of certain genes, certain sequences, and at the same time, it has the ability to unwind certain sequences so that those parts of the genes are exposed to transcription and ultimately gene expression. One classic example would be your green tea, which most of us consume during the daily basis. And your green tea has these polyphenols which can help with your DNA methylation directly or indirectly. And now consuming it on a regular basis will amend the patterns of your methylation, and it can also have an impact on turning off your oncogenes at the root level when it's done over and over again for a fairly long period of time. So this is exactly how your diet and your lifestyle can have a huge impact on your genetics, not by changing the DNA sequence, but by modulating what portions you want to express to enhance the quality of your life. And this is a very good example of how certain changes at the embryonic level, which results in embryogenesis, these changes at the embryonic level, the ones that you're born with, remains the same. You cannot modify because that's what results in subsequent embryogenesis and the division of labor at the fetal level. But during the adult life, you have the ability to write something which can be a permanent mark on your epigenome. For example, if by chance you have activated or you have expressed a certain oncogene due to an exposure to your biotic stress, or abiotic stress, which could be your carcinogens, your epigenome might be able to remember that, it might be able to foster those marks, and it might be able to implement that transgenerational inheritance where you see the same methylated groups repeat for the next few generations, exposing all of your subsequent generations to also the same condition. So what happens during that adult life especially with regards to your methylation, is variable. It's not permanent. It's not a permanent seal on the DNA, but it comes down to your consistent lifestyle practices where you create that epigenetic memory so that your generations, the subsequent generations, are able to remember those marks and effectively methylate the ones that's not supposed to be expressed. And some of the main important hallmarks of epigenetic control. So for the scope of what we do, we are mainly focused on the methylation, DNA methylation and the cofactors. For example, your SAMe, which helps in the form of supplements, it helps with methionine synthesis, and ultimately you need your methionine, your homocysteine to maintain your DNA repair mechanism, because your methylation cycle is intricately involved with the amount of methionine and homocysteine that you produce. When your SAMe is not functional, you do not generate the right methyl molecules, which can now shut off the expression of these bad genes. So which is why we mainly or predominantly focus on hyper or hypomethylating certain areas of your gene in order to make sure that you are not exposed to any of these chronic disease conditions, or if you are exposed, what can you do in the form of diet to minimize the impact of having certain gene expressions. And how do we do it? We do it using a precision medicine pipeline. So it essentially follows the step of collecting your DNA. We do the sequencing. Once we have the results, we evaluate your polymorphisms, conduct extensive bioinformatics to map these polymorphisms to something that's meaningful and resonate with the current status of your life. What are the lifestyle practices you're following, as a result of which what you could be exposed to. If there's a higher risk that comes from your genetic background, and if you've already diagnosed that during the formative years of life, it will be very, very easy or hassle-free for you to implement those changes since you've already discovered them at the early years of life. So you can implement with diet and lifestyle factors alone, just having these minute modifications, rather than having to go for something that's more chemical or treatment based. So this is the personalized medicine approach which we have incorporated for now. And how do we exactly do it? Based on your genetic profile, based on the sequencing results, we run the bioinformatics, and which is powered by a proprietary algorithm, and we are able to identify the ideal diet for you on a regular basis. And also we are able to identify certain important genes which, if present in a certain combination, can result in upregulation or downregulation of certain conditions. Classic example is the adipose gene, which I have mentioned here. Being in a certain genotype, since this is very much involved in the carbohydrate metabolism and appetite regulation, if it's going to be present in a certain variation, which is now not the normal variation, it can result in ease of gaining weight quite faster, or it might not be able to regulate your appetite. So in these cases, if you come up with a certain genetic background, what in the form of lifestyle you can do in order to make sure that you do not have that ease of gaining weight at a faster rate. And these are some of the examples of the DNA report, and we already have a booth here, so please go and have a look at the DNA report samples we have there. And the DNA report essentially tells you your predispositions, not the conditions you currently have, the conditions you may have if you do not have certain lifestyle factors in place. Like for example, when you look at the first map here, which gives you an insight on all of these vitamins, we all know we cannot produce these micronutrients. But what are some of the micronutrients you need on a regular basis is something our DNA reports will be able to tell you, that comes from your genetic makeup. And again, your predisposition to inflammation. So rather than looking at the end point of an inflammatory condition, a chronic inflammatory condition like cancer, we try to look at the root level. What are the chemokines or cytokines or interleukins you may upregulate, or you're already predisposed to upregulating, and now having a good control over your inflammatory background, your cascade, having an idea of that, you can go for an anti-inflammatory diet or a Mediterranean diet, which will help minimize the expression of your inflammation genes. So that's the end goal of what we are trying to do. Now, moving into the whole microbiome part, and this is a very nice diagram or an image that explains you need diversity when it comes to microbiome. You do not need one good strain in excessive quantity in your microbiome because that's not going to do the job. You need to have a good synergy of multiple strains, good strains, for you to be able to conduct your normal day-to-day processes, your metabolic processes. So a happy microbiome is when you have multiple strains, but in the right balance, not just one strain or the good strain in an upregulated fashion. And how do we exactly get our microbiome? So there are a myriad of things that contributes to the development, to the early development of the microbiome. One most important aspect would be breastfeeding, especially in the formative years of life, which is very, very essential to make sure that you have the first induction of the good bacteria in the gut, and that is what keeps multiplying and colonizing with time. And in the later years of life, what could hamper your microbial population is your antibiotics. One most important inflammatory disease condition that's associated with Clostridium difficile comes from excessive consumption of antibiotics. So when we do a genetic test, we are able to, the map here, we are able to identify based on certain genes that you have, whether you have the chance of upregulating certain microbial strains, certain gut microbiome strains like your Clostridium difficile. If you already have the chance of upregulating it, what are the prebiotics you need to take well in advance to make sure you do not have them in countless number in the gut? So that is what we are trying to do here. And this study that was conducted by, it was also published in Stanford's magazine, where there was a very beautiful correlation between how your microbiome can contribute to the pathogenesis of obesity. This sample was taken from identical twins, but the identical monozygotic twins, one of them was obese and the other one was normal. So now when there was a fecal transplantation into the mice here, there was one mice that had the sample taken from the obese individual that tend to develop, or that had the ease of developing obesity with time, and the other one which received the sample from the other twin who was normal, who was free from obesity, the mice seemed to be normal. So these differences, especially in this study, they were able to predict the close relevance of the dynamics of microbiome in resulting in obesity-like conditions, not just obesity, but also CVD or even neuroinflammation for that matter. And what are some of the genes involved which we are able to look at? So these are the three genes that I've mentioned here, but just to take one for an example, your FU2. So the FU2 gene that lines your gut allows the amount of sugars that can be produced at the gut. So now this stands as the substrate for many of these nutrients. Now, if you have this particular gene in a certain variation, which is called the SNP, which is the RSID, it could mean that there could be certain strains that can thrive over the other. For example, if there is a detrimental variation, if there is a certain variation associated with your FU2, you might have the tendency to upregulate the population of Helicobacter pylori. Now, if you have more of Helicobacter pylori, even if it's normal in certain cases, but upregulating the concentration with time can result in chronic inflammation or even cancer, adenocarcinoma in certain cases. So, which is why it's very, very essential to identify the presence of certain genes in the gut so that you're able to well predict what population survives and what population doesn't. And once we have the samples evaluated for what population could thrive in your gut, what atmosphere you're able to provide for certain strains to thrive, on the right hand side you can see the probiotics which we recommend for certain strains that you already have. Like for example, for your difficile, it's always good to have your gut repopulated with your Bifidobacterium bifidum and Saccharomyces boulardii. So these are some of the prebiotics which we can administer well in advance before you have the upregulation of certain strains in the gut. Now the bigger picture is the metabolic pathway. So this comes to the final section of the presentation where we have a unique metabolic pathway that we have generated based on the genetic makeup of every individual. So there's one section I have zoomed in just to understand or just to give an importance or the link between the presence of certain genes and the ability to upregulate certain microbial strains. For example, FU2 in a certain variation or a genotype can upregulate your Helicobacter pylori infection, the possibilities. So for this, the ideal thing that you would need to do is have the right good probiotics like your bifidobacterium. Now this is generated by a bioinformatics for every individual, which is unique. We are able to identify different aspects of metabolism, and at the root level most importantly, and even before any condition surfaces at the superficial level, which you get to know from a blood test for example, we are able to curate a targeted treatment complying with the principles of DNA methylation, so that in the long run you can prevent the dependence on any of these chemical medicines or mainly your medications. And I would like to end the presentation with the future directions. So an emerging field of microbiomics now tries to identify some of these short chain fatty acids which can result in inflammation in the long run. So some of the dietary molecules that you eat, for example choline, which comes from animal fat, when you consume too much of choline on a regular basis, your microbiome comes to act. They break down choline molecules into short chain fatty acids, and these short chain fatty acids, the presence at the gut-brain axis can gradually trigger neuroinflammation by activating your microglial cells, which are supposed to play a protective role, but because of the presence of excessive short chain fatty acids coming from your diet, broken down biomicrobiome, they can trigger neuroinflammation in the long run. So this is one thing that we are still trying to research. We are still trying to probe these aspects and find a targeted treatment for all of these conditions, mainly pertaining to inflammation. Thank you. Yeah.